In Chapter 1, we're going to look at these four big topics. We're going to take a look at levels of biological organization, talk about what that means. We'll talk about how life is defined by looking at what we refer to as the properties of life. We'll also take a look at some of the major themes of biology. And these are ideas that we'll actually come back to later in other chapters. So we're not going to spend a whole lot of time on those because they'll be revisited throughout the class. And then lastly, I'll briefly mention kind of science and the scientific method. And I'll talk about it just a little bit because we'll also be looking at it in our first lab as well too. So what do we mean by levels of biological organization? Well, biology is a huge field. Obviously, biology is the branch of science that studies life, what makes life happen, how do living things interact with each other. And so biology is really not actually just one field. Biology is a whole bunch of different fields. And these fields study life as it's organized at several different levels that go all the way down from the atom all the way up to all life collectively on Earth. And this outlines those levels here. The largest being the biosphere, which again looks at essentially all life on the planet as a whole, kind of the interaction of all the different ecosystems. That's what we would refer to as the biosphere. And of course, biology goes all the way down to the molecular level, even to the atomic level. So we could actually put a number 10 in here if we wanted. So biology takes a look at things all the way from molecules. We look at how molecules interact to form cell structures like organelles that are in the eukaryotic cells. We, of course, look at how cells themselves are produced and formed. And for multicellular organisms, we see how cells work together to produce tissues, and tissues in turn form organs. And when we think about cells, tissues, and organs, you don't want to think of a tissue as just multiple cells, and you don't want to think of an organ as just multiple tissues. It's a little more than that, because when cells connect and form structures together to actually create a tissue, that tissue can do functions that none of the individual cells can do themselves. And this is an idea in biology called emergence. It's basically kind of the same idea that the whole is bigger than the sum of its parts. So if we look at all the different cells in a tissue, each of these cells can do certain functions, but there are functions that the tissue can do that no one type of cell can do. So by working together, we get a synergy. We get something that the whole tissue can do that no one particular type of cell within it can do. And the same is true for an organ. An organ can do some function that no single tissue that makes up that organ can do alone. Again, that's an idea sometimes referred to as emergence. And we see it as we go from molecules that produce organelles and organelles working together with cells as well too. We see that basically as we move from each level of biology to the next. There's some new function gained that was not present in the constituent parts. So of course, we only see organelles in eukaryotic cells, and we only see tissues and organs in multicellular organisms. And an organism, of course, would be level number 5 here in our group. This would be an entire living thing unto itself. That's what we mean by an organism, an individual living thing. If we look at all of the individuals of a certain species within a certain area, then we call that the population. So organisms make up a population. And if we look at all of the different populations in an area, so basically looking at all of the different living things, well, then we call that the community. Okay, so population in biology means essentially exactly what you think it does. It's all of the individuals. But community in biology is maybe a little bit of a foreign term, so you want to make sure you're aware of what that means. A community means all of the populations. So it's basically all the living things of an area. If we also add in the non-living things that living things rely on, like water, air, sunlight, soil, then that gives us the ecosystem. So the ecosystem is the community plus the required non-living things that all the community members require. Then, as I mentioned already, if we look at how all of the ecosystems work together to kind of keep life on the whole planet sustained, then we're referring to the biosphere. So when we combine all the ecosystems, we get the biosphere. And so, you can pretty easily see this means biology is not just one field. It's not just one level of study. It's lots of different fields that expand over many different levels of organization. So another really key concept to take away from this first chapter is just what do we mean when we say something is alive? How do we define what is alive? And that seems kind of stupid at first, right? Because we all know that, a rock is not alive, but our dog is. We kind of just have this innate sense of what's alive and what isn't. But when you actually have to define what does it mean for something to be alive, it's really not as easy as you might think. And life cannot be defined with a typical kind of definition that you might find in a dictionary where we can, write it all out in just one sentence and okay, that's what we mean when something is alive. Instead, the best way we've come up with to define what is alive is a list of different properties called the properties of life. And in order for something to be considered alive, it has to fulfill every single one of these properties. None can be missing. If even one is left out, then whatever we're talking about isn't alive. So let's look at these properties of life. So properties of life include order. So we observe that every living thing is very highly ordered. It's very complex. It's very highly structured. Even the very simplest single-celled bacterium is not just this bag of stuff inside. It's very complex and very highly organized. So this order is definitely a characteristic that we see in all living things. Everything alive evolves. Everything has to adapt to its environment because the environment on Earth is constantly changing. And we'll talk about evolution a little bit in this intro chapter, just a tiny bit because it is one of the main themes of biology. We'll also look at evolution in an entire upcoming chapter in the course as well too. All living things have to have various mechanisms of regulation. They have to control themselves, and this is often referred to as homeostasis. Homeostasis refers really to kind of the internal regulation, how you control your body temperature, how you control your amount of blood sugar, how you control your heart rate. These would all be examples of some human homeostasis. But even when we get down to a single cell, even a single cell has got to regulate its internal processes to keep it all in sync so that it's functioning properly and to keep it in sync with its environment as well too. Every living thing also has to have energy. We'll talk about this in some upcoming chapters as well too. But every living thing has to have some way of obtaining energy and then processing that energy, converting the energy into just the right form so that it is a usable form of energy, so that there is a source of energy that the organism has to drive all of these other life processes that we would consider a property of life. Every living thing exhibits growth and development. And again, this is really easy to see in a multicellular organism like us humans, right? We know we're born small and we get bigger. And we also know that we've gone through a lot of changes. That's what we mean by development, right? We started out as a single cell, and then we were an embryo, then a fetus, then an infant, then a toddler, then we went through puberty, and then finally you're an adult. And so your physical self has changed quite a lot throughout that course. And that's what we mean by development, this ongoing change within an individual's life. So it's really easy to see in a multicellular organism, but it still happens even in single-celled organisms. Bacteria do grow. They get physically slightly bigger over their life cycles. And they do develop. They show changes in their morphology, their physical makeup, and they show changes in their gene expression as well too. I mentioned homeostasis. Homeostasis is sort of this internal regulation, and it does have to adjust the internal to kind of match the environment, but we have to have additional responses to the environment as well. When a predator chases a prey organism, the prey is going to run. Okay, we we respond to our environment at lots of different levels, from the molecular level to the conscious cognitive level. Gee, I think it's hot. I think I'll go inside where there's air conditioning. All of those responses to the environment definitely necessary to keep a thing alive. And of course, again, it happens even with single-celled organisms too. And then the last on the list here, reproduction. Right? Life has to continually produce new life in order for life to keep going. So these are still kind of evident. Yeah, of course, all living things do these things. But this is actually a really important list to keep in mind as you go through this class. As you look at the different topics that we cover, as you look at just even the chapter titles, you'll see that each one of those topics, each one of those chapters outlines some biological function that allows these properties of life to occur at the cellular level and at the multicellular organism level and at every level in between. In fact, every level of biological organization relies on these properties in order to establish those levels, and those levels all have to be present in order for a single living thing to have all these properties. So keep these ideas in mind. And when we look at a chapter on photosynthesis and respiration, you can say, oh yeah, this comes back to this property of life of energy processing. When we look at a chapter on mitosis and meiosis, cell division, okay, well, yeah, we have to do those in order to be able to reproduce. So essentially every chapter, every sort of biological function that we're going to study in this first general biology course can all be related to one or more of these properties of life. So this isn't some silly little list for you to memorize. Really think about this and really think, okay, what does it actually mean for something to be alive? You may have seen a list of properties of life in other classes that were slightly different, maybe organized a little differently, but it's essentially all the same idea. No matter how you kind of lay them out or define them, it's the same kind of core concepts. So we're also going to take a look at some of the major themes of biology, many of which we will revisit in this class. Some really don't get covered until general biology 2. But we're going to mention things like cell theory, evolution, expression and transmission of genetic interaction. We'll also take a look at how interactions occur. And this is something we'll see throughout the course as well too. Interactions are happening at every level. We can talk about molecular interactions. That's what we'll focus a lot on this course, frankly. And in general biology 2, you'll see things like interspecies interactions and ecological interactions. Biodiversity, also a really major theme of biology, but you really won't look at biodiversity too much until you get into general biology 2. So if you read through chapter 1 in your textbook or on your ebook, and you should, you'll notice that that chapter dedicates a lot of time to these different ideas. And again, these are all concepts that get revisited, if not through another chapter in this course, then certainly in a chapter in general biology 2. And I'm not going to try to spend a lot of time in this lecture except to just introduce these ideas in a broader sense. And really what you should take away when we're thinking about major themes of biology is, okay, as you go through general biology 1, and even next semester when you go through general biology 2, many of you will, think about how these themes are things that you see over and over again at many different levels and many different ways. So we're not going to try to get into all the details of these ideas here in this first lecture. We just want to introduce them basically as ideas to keep in mind, keep your eye open for as you move throughout the rest of the class. So one really important idea in biology is often referred to as the cell theory. And the cell theory is basically the idea that all living things are made out of cells. Okay, and there's kind of three tenets to the cell theory. And the first one I just mentioned. Every living organism is made up of cells. And the reason for that is that the cell is the most basic unit of life. So what we mean by that is no one has ever observed anything smaller or less complex than a cell that actually meets all of those properties of life. So the cell is the simplest, smallest, most basic structure that can actually support all of those properties of life and actually be considered to be alive. And then the third tenet is that cells arise from existing cells. Okay, basically spontaneous generation, the idea that living things just spontaneously pop up out of nowhere, doesn't really happen. Okay, life comes from already existing cells. Now, obviously, those existing cells had to get a start sometime several billion years ago, but that's kind of a one-off event. So cell theory, a pretty basic concept in biology, that cells are the simplest, most fundamental unit of life. They're the smallest things that can actually carry out all of these different properties of life. And if we look at the different kinds of cells, and we will do this later on in another chapter, we see that there are two basic types of cells for all life on Earth. A cell is either going to be eukaryotic or it will be prokaryotic. And the difference, the big difference between a eukaryotic versus a prokaryotic cell is whether or not the cell has organelles. So organelles are small membrane enclosed structures within a cell. Examples would include a nucleus or mitochondria, the chloroplast in a plant cell. Those would all be organelles. Prokaryotic cells do not contain organelles. Okay? Eukaryotic cells do. Eukaryotic cells are also going to be much larger, and because they have these organelles, and also because they have much more complex genes and genetics, eukaryotic cells, any which way you look at them, are going to be far more complex, far more complicated cells than prokaryotic cells. So if we look at what kind of animals have which kind of cells, well, anything multicellular is going to be a eukaryote. So anything multicellular, like plants and animals and multicellular fungi, are all going to be eukaryotes. There are also some single-celled eukaryotes as well, too. There are single-celled fungi, like yeast, and there are lots of different types of single-celled eukaryotes that we refer to as protists. These include things like amoeba and different little single-celled eukaryotes that you find swimming around in pond water, for example. So anything that's a eukaryote doesn't necessarily have to be multicellular, but everything that's multicellular is a eukaryote. Okay? There are no multicellular prokaryotes. Prokaryotes are essentially too simple to develop multicellular life. Prokaryotes are going to include things like bacteria and archaea. And this slide just kind of gives a comparison between a typical prokaryotic cell and a typical eukaryotic cell. And you can see the eukaryotic cell is much larger, and it's far more complex. It has these membrane structures inside, like the nucleus, that each have their own function, just kind of the way our own organs have a different function inside our body. These different structures called organelles each have their own specific function within a eukaryotic cell. So when we say prokaryotic cells are simple, that's really not saying that they're empirically simple. That means they're only simple compared to a eukaryotic cell. Even a prokaryotic cell is actually still extremely organized and extremely complex. They're not just little bags of goo that happen to be miraculously alive. Okay? They are very highly structured, very highly organized, and extremely complex. So when we say prokaryotes are simple, We really only mean relative to an extraordinarily complex eukaryotic cell. So what about a virus? Well, a virus isn't even made of cells. So a virus is not a prokaryote, and it's not even a eukaryote. And in fact, a virus is actually not a living thing. Viruses have some, but they don't do all of the properties of life. So the way a virus works is essentially it hijacks a cell. A virus is extremely simple. It is made up of nothing more than some type of genetic material, and that genetic material is going to be a type of molecule called a nucleic acid. This would be either DNA or RNA. Some viruses use DNA, other viruses use RNA. And to get that DNA or RNA into a new cell and take over that cell, a virus has at least a basic protein structure around it called a capsid. Some viruses are a little more complex, and they contain a membrane around the outside of the capsid called an envelope. But viruses compared to a cell are extraordinarily simple. They are not cells. And they are incapable of doing a lot of those properties of life. In fact, a virus can't actually reproduce itself. What a virus does is once it infects a cell, its genetic material, its nucleic acid, reprograms the cell, and the cell actually starts producing and releasing more virus. So a virus doesn't actually reproduce itself. It takes over a cell and directs the cell to make more. A virus also doesn't do anything to get its own energy. It basically just uses the energy that the cell got. A virus does do some properties of life, however. They certainly undergo what we could argue is development, because through a viral replication cycle, we see changes in gene expression, and we can actually see changes in that gene expression as a response to what the cell is doing. So we do see some response and we see some development. Again, change in gene expression can constitute development. So viruses do some properties of life. And probably the one that they're best at is evolution. Viruses evolve very, very rapidly, far more rapidly than cells do. So viruses do some properties of life, but not all of them. In fact, not even close to all of them. There's a whole lot that are missing. So even though they're pathogens, and even though we very commonly refer to a virus as alive or dead, meaning in reality, is it infectious or not, none of them are actually alive. Okay, they don't do these properties of life. They actually don't constitute a living thing, even though they're still a pathogen, even though they can cause disease. They don't meet those properties of life, and they're non-living. So like I said, sometimes the idea of what's alive and not seems very almost kind of stupid to even ask. Of course, the tree is alive. Of course, the dirt that the tree is growing in is not. But when you get to something like a virus, you actually have to think about it a little. So we mentioned, again, we would look at several of these themes. And again, cell theory is one big idea. And of course, when we look at viruses, that ties in with cell theory, right? It's only a cell that can do all of these properties of life and be considered alive. A virus is not. Okay. Another big theme that we see in life that actually viruses, even though they're not alive, still do, is that transmission and expression of genetic information. So again, what we mean by transmission is passing genes on from one generation to the next, right? We pass down our genes to our children, and viruses pass their genes off to the new viruses that the cells make for them. And what we mean by expression of genetic information is actually using those genes. Okay, if we have a gene for a particular trait, then when we express that gene, that creates that trait within us. Okay? And we'll spend several really important chapters looking at what genes are and what gene expression is and how all of that works. And we're going to see that for all cellular life, the molecule of inheritance, the molecule that stores this genetic information, is going to be DNA. So whereas viruses use RNA sometimes as their genetic material, all cellular life uses DNA. DNA is short for deoxyribonucleic acid. So we'll have some upcoming chapters where we look at DNA structure, at how DNA is replicated so that we can pass it down, at how our chromosomes are divided out when their cells divide, and at how genes actually work. But the most important basic idea to have about DNA, this early on at least, is the idea of what DNA's function is. The function of DNA is to store information. That's what it does. It is information storage. And we'll see that it is very literal information storage. I don't mean it's metaphorically storing information. It's storing information, just like the notes that you're writing right now when you listen to this lecture is storing information, just like all the ones and zeros on your computer's hard drive is storing information. DNA literally stores information. And this information is what makes us who we are and what we are. And when a cell uses that information, that's what we mean by gene expression. Okay? And we're going to see that typically the function of a gene is to store the information that tells a cell how to make a type of molecule called a protein. And we'll see that it's proteins that actually carry out all of the biological activity in a cell for the most part. So when we think about how do we develop, how do we change? How do we grow from a single cell to an adult? How do we control our interaction with the environment? How do we regulate ourselves internally? Well, ultimately, all of that information needed to do all of those properties of life is all encoded in the DNA. So gene expression, again, is using that information, putting it to use to actually create a functional living thing. And we'll have some really important chapters where we look at that coming up. So again, all that information that we need to produce a human, in our case, is all stored within DNA molecules. And it's not just information on how to grow. It's also all the information on how to do all of these properties of life. This is what ultimately gives the information we need to adapt, to respond to the environment, and it's also going to be a basis as it changes of evolution as well too. So as we go through this course, as I mentioned before, and even when you go on and look at the topics of General Biology 2, where we look at organs and organ systems and the upper end of those levels of biological organization, you're going to see lots and lots of interactions. Interactions occur everywhere in biology, from molecular interactions that allow gene expression to happen within a cell, to cell-cell interactions, organ interactions, tissue interactions, interactions between organisms, interactions between populations, between ecosystems. I think you get it, right? It's every single one of those biological levels is all constructed on different sets of interactions. And so interactions are a big driving force in all biology. And we see these interactions in different ways at different levels, right? So at the ecosystem level, we would see interactions between organisms, for example. And we even see interactions between ecosystems at the level of the biosphere. So we have to see interactions even at the molecular level to allow these bigger interactions to occur. So when we're thinking about how do animals get the energy they need from their environment, well, we do so by eating things. And ultimately, it's the biochemistry of those food molecules storing energy that gives us the energy that we need. And those are the type of interactions that we're going to focus on in this course. This course is going to look more at cell biology, molecular biology, a little bit of biochemistry, genetics, that level of interactions. And one thing that we'll see later on in this chapter is the idea of feedback regulation. And we can see this in some degree at every single biological level, but where it's often most obvious is kind of at the molecular level. And what we mean by feedback is a regulation in which the end result of some biological process, okay, the output, the product that some biological process makes, is what actually controls that process. So, for example, imagine one of your cells needs to make a particular type of molecule. So we're going to learn that the way cells make anything is the use of enzymes. Enzymes are proteins that catalyze biochemical reactions. So let's imagine one of your cells is undergoing some biochemical reactions to make a particular molecule that the cell needs. Well, that molecule is going to actually control its own production. And that's what we mean by feedback. The end product of some process is actually the component that ultimately is controlling the process. And this can happen in two different ways. It can happen as either positive feedback or negative feedback. In positive feedback, that product that's made will act as a chemical signal. The product acts as a chemical signal that stimulates even more of the product to be made. So it's kind of a snowball effect. A little bit's made, so let's keep making more. That's actually a little uncommon. More commonly is feedback regulation that is referred to as negative feedback. And in this case, that molecule at the end of some process turns down the making of that molecule. So basically, once you've got enough, then the cell would quit making more, right? So negative feedback. So usually the way this works is at the end of the process, that chemical goes back to the beginning and turns it off. And as an example, we can look at the way your body produces insulin. So insulin is the hormone that triggers your cells to remove glucose from your blood when your blood glucose levels get high. And we all know that if this regulation doesn't occur, that's diabetes, when your blood glucose is misregulated and becomes too high in concentration. So let's take a look at kind of how this process works, and we'll be able to use it as an example of when we would see negative feedback. So let's imagine you just ate a big donut or something that's good and sugary, and after you digest that and absorb all that sugar, well, your blood glucose level would be a little higher, and that's completely normal. And your body's going to respond to that so that it doesn't stay too high too long, which would become dangerous. So there are cells in your pancreas which can detect your blood glucose level. And when they detect that blood glucose level is a little too high, a little out of balance, not quite in homeostasis, then those cells make this protein called insulin and secrete it into the bloodstream. Insulin is a hormone, and so it's going to be circulated throughout all your blood, throughout all your body, but it only affects certain cells in your body, specifically certain cells in your liver and in your muscles. And when the insulin reaches those cells, these cells are stimulated to start taking glucose out of the blood and building it up into larger sugars for storage. Okay? So that's how we regulate our blood sugar. The insulin triggers cells in our livers and muscles to take sugar out of the blood and build it up into bigger sugar molecules for storage. So the result of this is that the glucose level goes down. And once that glucose level has gone down, well, then that turns off the production of insulin. Because if there's no high glucose anymore, the insulin production will stop. So again, what is the end result of this process? Well, the end result is to drop our blood sugar. And when the blood sugar is dropped, well, that turns the process off. Okay, that's negative feedback. The end result of some process is what is controlling the process. And when that end result is turning off the process, that's negative feedback. Positive feedback would be if the end result kept the process going more and more and more. Positive feedback is kind of rare. But I will give you a good example of positive feedback too, just kind of so you give an example. During childbirth, the mother begins to release a hormone called oxytocin. It does a lot of different functions during childbirth, but one function is to induce labor, to induce the muscle contractions that actually push the baby out. And oxytocin is controlled by positive feedback. So once the mother starts producing a little bit of oxytocin, that oxytocin stimulates production of more oxytocin, and that stimulates production of more oxytocin until finally the baby is delivered. And that's an example of positive feedback. Positive feedback is going to happen in cases where you don't want things to quit until, of course, they are completely finished. Now, once the child is delivered, there's other mechanisms that do eventually shut down the oxytocin production. But positive feedback is where the end product stimulates more production of that product, and negative feedback is when the end product turns off more production of that product. But in both cases, it's feedback because the product is controlling it. It's feeding back to regulate it. And I went ahead and mentioned this in kind of our big themes because it shows interactions, right? We have interactions with these insulin cells in the blood to make this, to trigger this process, to trigger this response. We have interactions with the insulin that's released with the muscle cells. There's interactions occurring everywhere. And interactions and feedback interactions are huge ways that we control and regulate all of these biological processes. And we'll see lots of interactions like that as we go throughout the course. I've got a couple of animations that demonstrate negative feedback and positive feedback. I'm not going to play them here in this video, but you should play through them when you look at them as you study the PowerPoints. We mentioned biodiversity also as a big theme of life. And again, you will look at biodiversity a lot in general biology too. The reason we have general biology in 2 courses is at the depth that we look at it, we couldn't fit it all into one. So biodiversity is really a topic for general biology 2 more than general biology 1. But I will just mention, how do we wrap our heads around all that diversity? So there's almost 2 million species that have actually been identified, things we have written in the books, things we have sort of officially given a name, and we identify hundreds, if not thousands more every year. And this is probably a drop in the bucket of what's actually out there. And in actuality, there's probably 10 to 100 million different species. And actually, I think these numbers are just for eukaryotes alone. So if we were to include prokaryotes, it would be many times over. So there's a ton of different living things on Earth. How do we keep track of it? Well, we can't keep track of 100 million different individual species. So we have to have some way to categorize them, to group them, to be able to wrap our heads around it. And to do that, we use what's called taxonomy. Taxonomy is the name for the classification system. And it's going to divide all living things into groups. These groups are called taxa, and that's where we get the name taxonomy from. So an individual group is called a taxon, and taxa would be the plural. And this divides all living things into these major groups: domain, kingdom, phylum, class, order, family, genus, species. And where necessary, we even add some groups in between. Like we may have something called a superphylum, or a subphylum, or a subclass. So these aren't the only taxonomic divisions. Sometimes we have to add some extra in there to still keep track of everything. But probably everybody's familiar with taxonomy already. But just to give a really brief overview, we start with this group called a domain, and then domains are subdivided into kingdoms. Kingdoms are subdivided into phylums, and so on and so on and so on, until finally you get down to just a single type of organism, and that, of course, is called a species. So let's kind of look at an example here a little bit. First, let's think about the domains. Well, every living thing is first divided into one of three domains, and these are bacteria, archaea, and eukarya. So 2 out of these 3 domains, bacteria and archaea, are all made of just prokaryotes. And when you study prokaryotes, one thing you'll be told right away is that bacteria and archaea are very, very different from each other. So it's not like, well, archaea is just a kind of bacteria. Okay, that's not true at all. There are 3 very, very distinct basic branches of life, and those are the 3 big branches. Bacteria, archaea, both of those prokaryotes, and then so every kind of eukaryote all goes into this domain called eukarya. And that's going to include every kind of multicellular organism, because all multicellular organisms are eukaryotes, plus a whole lot of single-celled eukaryotes as well too. So let's kind of look at how we would classify something like the American black bear, just as an example. Well, first of all, this bear is a eukaryote, so it would be put into the domain of eukarya. And again, this is big and broad. This is going to include things like plants, fungi, even some single-celled things like amoebas that live in ponds, paramecium. Okay, so very, very big, huge division. Then we're going to take this domain of eukarya and we're going to divide it up into kingdoms. And there's going to be the animal kingdom, the plant kingdom, fungi, and a whole bunch of different kingdoms that make up these single-celled eukaryotes called protists. So for the bear, obviously it's going to go in the animal kingdom, or kingdom Animalia. Then we're going to divide that animal kingdom into phyla. And a bear would go into a kingdom called Chordata. Chordata includes all of the animals that have vertebrae, plus some invertebrates too. So a bear obviously got a backbone, that's a vertebrate, so it would be considered a member of the phylum Chordata. We then split the phylum into classes. And so of the different classes, bears would be in the class called Mammalia, which would be mammals, right? Other classes would include reptiles, amphibians. Then we would divide the class, mammals, into different orders. And within the mammals, a bear would go into an order called Carnivora, which would include all of the carnivorous animals, all the ones that eat almost exclusively animals. Within Carnivora, the order, we would then subdivide it into the family that includes most of the bears, Ursidae. Then we would divide it into genuses, and it would go into the genus of Ursus, which includes polar bears and grizzly bears as well too. And finally, we would give it a species name, Americanus. So the scientific name for the black bear is Ursus americanus. And the scientific name comes from the genus and the species. Okay, for us humans, you've probably heard, we are called Homo sapiens. We belong to the genus Homo, and our species name is Sapiens. So this is where the scientific name comes from. So taxonomy does 2 really important functions in biology. First, it puts things into groups so that we can kind of make heads or tails of all this biodiversity, right? We can't think of 100 million different eukaryotes. However, we can start dividing the eukaryotes into groups. And by studying each group, then we can kind of begin to wrap our heads around all the biodiversity that's out there. So it allows us to just, with our finite, limited human brains, begin to understand all this biodiversity. The second really important thing that taxonomy does is it gives us a systematic naming system. So the scientific name of something is the genus and the species after it's been put through all of this taxonomic sorting. And anytime you see a scientific name written, it should be written like this. The genus should always be capitalized, and the species should always be a lowercase letter. And since all of these names, as you may have noticed, are kind of Latin, or sort of fake Latin at least, this is considered a foreign language. This is considered to be a Latin name. And in English, anytime you write a word that's in a foreign language, you italicize it. Okay, now there's examples or exceptions to this, which are words that are commonly used in English, like taco, right? You don't need to italicize taco, okay? But you do need to italicize a scientific name anytime it's written. So a properly written scientific name will be in italics, capitalized genus, lowercase species, and sometimes you'll see an abbreviation where they'll just use the first letter. So they may call this U Americanus. So there's this incredible diversity, right? Biodiversity is really too much for us to comprehend. The human brain really can't wrap our heads around it. But no matter how diverse two organisms we pick, we can still find commonality between them. From the most complex animal to the very simplest bacterium, we still find amazing unity within all of that diversity. And that's because all life has some common origin, some initial beginning life that we have eventually evolved from. And where we see that university most, that unity most, is at the level of DNA. We're going to see that DNA, even though it can be very different from a bacterium, a prokaryote versus a eukaryote in some important respects, that same genetic code works the same way in essentially all organisms. That's kind of the big unifier. So the more closely we get down to the molecular level of biology, that's where we see more and more commonality, more and more similarity. So again, we have this big tree of life as the original living things have developed and diversified to create this incredible amount of biodiversity that we see here. And we can see this in a lot of different ways. Where it was originally found, where it was originally observed, was of course fossils. So the fossil record kind of traces how things have changed over time. But we can also see changes even in things that are still alive. We can use comparative anatomy to kind of see how diverse things still have a relationship. And an example of that is shown here. If we look at a bat wing, the flipper of a porpoise, or a human arm, these are very different structures, right? This bat wing is for flight. The porpoise flipper is for, paddling in water. And a human arm really isn't used to move us around. It's used for grabbing things and grasping things, right? We don't walk with our hands and arms anymore. But even though these have actually diversified to do three very different functions, they're all still coming from mammals. And we can still see common ancestry as common mammals in the bone structure. Because if you look at any of these structures, we see a pattern. We see one bone, two bones, a whole bunch of bones, like in her wrist, and finger-like bones. Okay? We see that in the porpoise flipper. We see that in the bat wing. And not only do we see this across all mammal limbs, we also see it in reptile limbs. We go back even farther, and we see it in amphibian limbs. And we can go back even farther than that, and we can see it in lobe-finned fish. So by comparative anatomy, we can kind of trace that commonality. And of course, we still see it most if we begin to trace DNA sequences. It's at that molecular level that we see the most unity and the most commonality, but we can still see it even in gross anatomy as well too. So this brings us into one of the big themes of biology, which is evolution. And we'll spend an entire chapter on evolution. But the only thing I want to point out now is that we often take multiple ideas and sort of all blend them together and confuse them and call all of that together evolution. And evolution actually has a very specific meaning. And we're going to talk about that a little bit now. But one thing that we typically associate with evolution, or the person whom we associate with evolution, was Charles Darwin. And if you ask most people who came up with the idea of evolution, well, they would say Darwin. And that's actually incorrect. And this speaks to what I mean about we confuse what evolution is. So, what Darwin came up with was not evolution, he came up with natural selection. Evolution predated Darwin by a couple hundred years. People knew long ago from finding fossils that things were changing over time. That basically is evolution. What Darwin came up with was the reason that things change, and the main reason that things undergo adaptive change. Okay, and that is natural selection. And he published that in 1859. But people knew way back into the early 1700s and even the 1600s, probably through finding fossils, that evolution was occurring. So, why do I mention this? Well, I want to point out that natural selection is different from evolution. And also, speciation is different from evolution. Yet we often take natural selection, evolution, speciation, and kind of blend them all together and call it all evolution. But in this class, we're going to identify some very separate definitions for them. So when we get to our chapter on evolution, we're going to have a very specific definition for it. And here's the definition. Evolution is a change in the frequency of alleles in a population from one generation to the next. Well, if that's not clear, the frequency of alleles is the gene pool. Allele means a form of a gene. Right? We have, for example, alleles that affect our eye color. Do you have blue eyes or brown eyes or green eyes? Different versions of those genes that control eye color would be alleles. So what evolution is that we see from one generation to the next generation a difference in the frequency of those alleles in the population. So for example, if we used the human population of Tennessee as our example population, and we looked at eye color, and we counted right now how many people have blue eyes versus green eyes versus brown eyes. We would be counting the frequency of those alleles, essentially. And when we looked at the next generation, we would see it wasn't an identical number. It would have changed a little bit. Okay, that's what we mean by evolution. Okay, we don't necessarily mean that it's an adaptive change. It doesn't have to be a biologically important change. And we're not talking at all about what caused that change. We're just observing that a change in the gene pool happened. That is all evolution really is. Now, is eye color an important biological change? No, it's not. It doesn't matter at all. It doesn't make us any more healthy or less healthy. It really has no effect on us at all. So not all evolutionary change is important. But a lot of evolutionary change can be important. It can allow for a population to become better adapted to its environment. And if enough evolutionary change accumulates, then the species does become something different. And that's what we mean simply by speciation. So speciation is when enough of this change has occurred that something becomes different than how it started out. Okay, so speciation is a result of evolution. So what then exactly is natural selection? What was it that Charles Darwin came up with? Well, natural selection is actually a cause of evolution. And we're going to see when we look at our evolution chapter that there's a lot of different causes of evolution, including just chance, just dumb luck. But natural selection is a particularly important cause because it does actually tend to cause change that is adaptive. It tends to make things become better suited to their environments. So what we mean by natural selection is this process, this cause of evolution. And what happens during natural selection is we have this population. And within that population, there's going to be some of the organisms are going to have certain characteristics that make them a little better adjusted, better adapted to their environment than some of the others in their population. So those individuals who have these advantages are going to be able to survive better, and more importantly, they're going to be more successful at reproducing. So they're going to have more offspring. And if they have more offspring, that means more of their genes are going to be getting passed on to the next generation. So we're going to see more of those alleles than other alleles. So this has caused a change in allele frequency, which is evolution. So if something has some advantage that allows it to survive better and be more reproductively successful, we'll see more of those genes that gave that advantage in the next generation. Okay, so that is natural selection. So you can see natural selection causes a change in the gene pool, but it is not itself evolution. It is a cause of evolution. And that's what I mean by we want to have a clear definition of evolution, and then we want to separate the causes, like natural selection, from the effects, like adaptation and speciation. So in our evolution chapter, we'll look at many other causes of natural selection, and we'll look at several different ways in which speciation occur. And we can, of course, point out again that not all change leads to adaptation, and certainly not all change even leads to speciation. Sometimes evolutionary change is just change that isn't really biologically important at all. Okay? But understanding or kind of separating away the concept of bare bones, what really is evolution, separating that away from causes and effects will really give you a clear understanding of the processes involved. So the last thing that I want to mention here in chapter 1, and I'm not going to talk too much about it because you're going to be looking at your entire first lab on this as well too, is really what do we mean by science? And essentially, science refers not to all of the fields that we have. We don't mean what's in the biology book, what's in the chemistry book, what's in the physics book. We don't really mean the bodies of knowledge when we say science. We actually mean a way of learning things. So science is a way in which we learn about the natural world. Okay? And the way that science learns all this stuff is the scientific method. So to kind of give you just a comparison, there's other ways of learning, right? You can also learn about the world through theology. You can learn about the world through philosophy. You can even learn about the world through experiencing and making art. Those can all kind of inform us different things about our world. But when we want to learn about the nuts and bolts, about matter and energy, and about natural processes, about the physicality of the world, the best way to learn about it is through science. And that's because science has this particular way of learning called the scientific method. And what you're going to see when you go through your first lab is exactly what the scientific method is and exactly how the scientific method works. And in a nutshell, the way that we learn about science is that we have this idea called the hypothesis, and we don't know if this idea is right or not. It's our best explanation that explains some aspect of how the physical world works. That's a hypothesis. And we don't know if it works or not. We want to find out, are we right or are we wrong? So what science does is it puts the hypothesis to a test. It conducts an experiment to test, is the hypothesis right or is the hypothesis wrong? And that's how science learns. You may think, well, doesn't everything learn kind of that way? Well, actually it doesn't. And science is very special because science isn't trying to find evidence to prove the hypothesis is right. Science isn't creating an argument. It's not building a case. It's legitimately putting this idea to the test. Is it right or not? And when you do an experiment, if your idea is wrong, you want your experiment to show that idea was wrong. And then you go and change the idea, and then you test your new idea. And that's basically how the scientific method works. You've seen the scientific method described to you several times in your educational career. You've probably never seen it in exactly the same steps, but this is kind of the most basic way of looking at the scientific method. We've observed something. It's made us curious. And we've come up with a logical, rational, very well-educated idea of how this might happen. And we call this explanation a hypothesis. Okay? This is how we think, whatever our observation was, this is what we think is going on. Okay? But we're not sure. We want to know how it works. So we make a prediction. If this idea is true, then we should see that. And we use that prediction to make an experiment. Okay, so the experiment is going to test the prediction. And if the prediction comes out to be true, then we conclude that the hypothesis may be right. If the prediction did not come out to be true, then the conclusion from our experiment would tell us that our hypothesis is wrong. Okay, so that's the basic idea. And in this first lab, we'll kind of talk about that more, and we'll kind of use a little silly kind of example to kind of step through these steps, sort of at a slow pace, one at a time, to really get an idea for how this works. But it really is a very special way of thinking about things, and really only science actually does this. We don't see this in philosophy. We don't see this in theology. We don't see this in art. Okay, science is very unique in that it takes its ideas and it puts it to a real test. Is this true? Is this not? I'll give you another example of a way that we use to try to figure out what is reality without using the scientific method, and that is actually a court of law. And this isn't to say a court of law doesn't work as well. Okay, what a court of law is trying to find out is very different than what science is trying to find out. And science often won't work to settle a court case. It can't work. There's no way to test what happened in the past in most cases. So a court of law works by having two sides, right? A plaintiff and a defendant. And they're each building cases. They're creating an argument, and they're trying to convince a jury that their argument is right and the other argument is wrong. And that is a fantastic way to address the kind of questions that would be brought up in a court of law. But the kind of questions that science is answering is very different. We're trying to answer fundamental questions about how does nature operate? What makes photosynthesis work? What makes a cell alive? Those are the biological questions, right? And so these are questions that we can put to a test with an experiment. And when we put that question to a test, it's not like a court of law where we're trying to build a case to prove ourselves right. We're actually trying to find out that if it's wrong, we want to know it's wrong. And that's what is really unique about science. It's very rigorous. It's very honest with itself when it's done the right way, and it is very unique in that regard. And this methodology, the scientific method, that's it in a nutshell. You have a hypothesis, and you're going to put it to a real test by performing an experiment to check and see, is your hypothesis right or not? It's not ideological. We're not going to come up with an idea and find every which way we can to convince everybody else it's right. We are going to take this idea and we are going to torture it to make sure it really is correct. So again, that is a hypothesis, this tentative idea. We can use predictions to figure out how to make an experiment, and we'll talk about that more in lab, so I'm going to kind of leave that there. But to make an experiment a good experiment, to make sure that it's reliable, we're going to talk about in several of the labs that we do, not just the first one on the scientific method, about things that can be changed in an experiment. So something that can be changed when you do an experiment is called a variable. And in your experiment, there are two really important variables. These are the independent variable and the dependent variable. And this is so important, I'm going to talk about them here first, and you'll hear about them in labs, as upcoming labs as well too. But the thing that you're trying to test in an experiment, so when you're doing an experiment, usually you're going to change something and then see what effect does changing this have. So the thing that you're changing, the thing that you're actually testing, is called the independent variable. And you're going to see the effect of this independent variable by measuring something, by monitoring something. And that thing that you're measuring or monitoring is the dependent variable. Okay, so here's a little example. Let's imagine you're a baker and you wanted to test which kind of flour works best in helping my cookies rise. And you wanted to test, well, is it better to use cake flour or is it better to use all-purpose flour? So what are you going to do? Well, you're going to make up some cookies with cake flour and make up some cookies with all-purpose flour, and you're going to see which one rose the most, right? Pretty simple experiment. So let's think about these variables. Your independent variable would be the thing you're testing. What kind of flour did you use? Did you put in the cake flour or did you put in the all-purpose flour? Okay, that's the thing in your experiment that you are specifically testing, the thing that you are changing on purpose to see what effect does this have. So that would be the independent variable. Your dependent variable is the change you're looking at to see the effect. So in this case, it's how high did the cookies rise, right? That would be our dependent variable, right? The dependent variable depends on the independent variable. How high the cookies rose depends on which kind of flour we put in. Okay, those are kind of the basic ideas of variables in an experiment. Now, those are not the only things that could have changed, right? We could have changed how much water did we put in? What temperature did we cook them at? Did we use margarine or butter? Those are all variables, but we don't want those variables to affect the experiment. So when we grew our batch of cookies with the cake flour, and then we baked. When we baked our batch of cookies with you can tell I'm a biologist, right? When we baked our batch of cookies with the cake flour, and when we baked our batch of cookies with the all-purpose flour, the only variable we allowed to be different would have been the independent variable. We would have kept all other variables the same. We would have used exactly the same oven temperature. We would have used the same amount of water. We would have used the same kind of butter, the same amount of butter. We wouldn't have had anything else different. And that way, when we look at our dependent variable, how high did the cookies rise? We know that any difference must have been due to the independent variable, the thing we wanted to test. So this is what is referred to as a controlled experiment. A controlled experiment uses a comparator. Okay, in this case, our batch of cookies with the all-purpose flour would be a control to see what effect did using cake flour have. Well, cake flour made it grow taller. Well, what about the water? Did the water have an effect? No, it didn't, because we used the same amount of water. Okay, that's called controlled. We have controlled for every other variable except the independent variable. Every other variable is exactly the same between our experimental group and between our control group. Okay? So we use controls in order to do that, and an experiment that has controls is called a controlled experiment. And again, the whole reason for using these control groups is so that the only thing that's different is the independent variable. So any difference you see, you know, was because of that change in the independent variable, and that tells you your experiment is giving you a reliable result. And there's not something else that is accidentally causing some effect and giving you a misleading result and therefore a misleading conclusion. So whenever experiments are done, we always do a controlled experiment. We'll talk more about controls in that first lab on the scientific method, and we'll talk a lot about controls in upcoming labs as well too. One way also to make scientific experiments reliable, to help eliminate bias, is to record quantitative data instead of qualitative data. Qualitative data is descriptive data. So we would say, oh, the cookies rose a lot versus a little. Well, what does that mean? And that actually has a lot of judgment on us. It's our opinion of if they rose a lot or a little, right? We would want quantitative data. We would actually want to take a ruler or a caliper and actually measure how much did these cookies rise, measure how much did those cookies rise. We're not going to argue whether it's a lot or a little. We're going to have a number on it. So data with numbers is called quantitative. Descriptive data is called qualitative. And if you describe something, that gives room for you to put your opinion in there. And science doesn't work on opinions. It doesn't work on our judgment. We don't make judgment calls in determining experimental outcomes. We want numbers that we can compare to determine our experimental outcome. So we use quantitative data and we do controls so that we know that what we think we're testing, the independent variable, is what's actually producing the result. And that makes experiments reliable. That's how experiments put us on the right track rather than giving us weird results that lead us down the wrong conclusions. So we're going to talk a lot about controls. We're going to talk a lot about what's the independent variable, what's the dependent variable as we do labs throughout the course. And again, a really important concept about understanding how science works is that the goal of the scientific method is to use an experiment to test the hypothesis, okay? To test, is it correct or is it not correct? An experiment is not there to try to prove your hypothesis is right. An experiment is an honest, rigorous test so that if you're wrong, your experiment should tell you you're wrong. Okay? It's not gathering evidence to support a particular idea. And science always remains open to the idea, no matter how many experiments you've done, no matter how well supported an idea is, if somebody comes up with really good data, with extremely good experiments that says, hey, you know this thing we've thought for 100 years, maybe it's wrong, well then science will admit, yeah, that was wrong, let's change it. Now this doesn't mean science is flip-floppy. It takes a whole lot of good data to overturn a really well-established idea. Science doesn't flip-flop, okay? But it is open to being changed if we find out something really is wrong. So science never would ever say that it has proven a hypothesis, okay? Because we always want to be able to change it in the future if in fact we did find out it was wrong. But when we do have these ideas where we have tested them over and over again, and we have essentially had a whole bunch of different, very well tested, very well supported hypotheses that all relate to each other and create this big idea that we essentially would say we're as confident as this being true. We would never call it proven, but essentially we think it's probably proven. That is a theory in science. We use the word theory in science very differently than we use the word theory in regular old English conversation. In regular old everyday English, we would use a theory to mean it's just a guess. And a theory in science is not just a guess, right? Somebody asks you a question and you have no idea, you're just going to throw a guess out there. You might say, well, I've got a theory, right? So that is not what theory means in science. It's actually kind of the opposite. So in science, a theory is this big idea. It's not just a hypothesis. It's essentially a whole bunch of related hypotheses that have been very well supported by a huge, huge number of experiments. Okay, again, if something has essentially been proven, although we don't really want to say proven in case it might happen to be wrong, but essentially we really, really think we've got this right, that's what we call a theory. Okay. Again, it's not just a single hypothesis. It is a big idea, but it is a big idea that is fully accepted by lots and lots and lots of experimental evidence. Okay, so the theory of gravity, the theory of evolution, cell theory, These are things that through lots of science, lots of experiments, we essentially said, yep, we're almost certain we have not messed up. These things we hold to be definitely true. Okay, so theory and science, a little different than the way the word is used in regular everyday language. So, as I said, as you go through your first lab, it will also kind of revisit these ideas about the scientific method a little more thoroughly than this quick run through in our first lecture for the first chapter. Be sure to focus on the ideas of the levels of biology and the properties of life. And also, as I mentioned already, we will revisit those kind of big themes in science, like interactions and evolution and biodiversity, if not in other chapters in this course, then in the case of biodiversity, with many, many different chapters when you get to general biology too. So this first chapter really has kind of three big ideas to focus on now, and then it introduces other ideas that we'll see later. So the big ideas to focus on now is what is science and the scientific method? What are the properties of life? What are all of these different levels of biological organization? So this is a pretty short, pretty simple first chapter. And I encourage you, as you're getting underway in this class, please go ahead, read chapters 2 and 3 to do a little chemistry review if you need, and then start in on the next chapters for the course, which are chapters 4 and 5. Don't wait till next week. Go ahead, start on those now. Those can be kind of challenging. So look through those, do a little chemistry review, and be sure to contact me by email or to set up a Zoom meeting if you'd like a little one-on-one explanation of stuff. If you have any questions about any of these ideas.